Okay, so protein synthesis happens in two stages. First, you've got transcription, then you've got translation. So remember, transcription is taking something and just writing it again in the same way. So you are taking DNA and you're rewriting it to RNA. The same kind of nucleotides. Okay, so we just take it and we write it. Translation is taking it into a different language. So translation, we're going to take that RNA and we are going to turn it into a polypeptide full of amino acids. Okay, so you're taking it into a completely different language. So we're going to start by looking at the first stage of transcription. So transcription happens in the nucleus. Okay, so in the nucleus, you've got your DNA. Okay, so if this is my nucleus in my cell, there's my strand of DNA that I want. The section of DNA that codes for the protein is the gene. So this specific gene is going to unravel and we're going to use one side of it as a template. Now that template is going to be copied into RNA. Okay, so we're going to have RNA along, and every base is going to complementary base pair with another one. So T always matches with A. A in RNA always matches with U, and C always matches with G. So that's going to keep matching, and we are going to keep making a sequence. So G with C, G with C, A with U. C with G, A with U, and G with C. And you're going to keep going along, matching the whole strand, and this will become an mRNA. So this is your mRNA, and the M stands for messenger. So the messenger RNA is going to be made in the nucleus through the process of transcription. Okay, we're going to join the sugar phosphate backbone together with the help of the enzyme RNA polymerase, and we're going to have one solid strand of mRNA, and every 3 letters is what's going to code for an amino acid. So those 3 letters in the DNA are a triplet. The 3 letters in the mRNA are a codon. Okay, so each one of those will code for one amino acid. So there will be 1, 2, 3, 4, etc. as we go along for every 3 bases. So once it's been made in transcription, then that's going to move out of the nucleus and into the cytoplasm. It is going to go straight into a ribosome. In the cytoplasm, in our ribosome, it's going to surround the mRNA, and this is going to be translation. Now, for translation, you have little free molecules that look like this, in the shape of a T, and these are called tRNA. The T stands for transfer. So these tRNAs are made of nucleotide bases, and they're held together by hydrogen bonds. Okay, on the one end, they have 3 bases that will match these 3 complementary base pairing, and that will be called the anticodon. Okay, so the 3 bases at the bottom are the anticodon, which matches the codon. And on the other end is the site for picking up the amino acid. Okay, so the tRNAs, the transfer RNAs, will go into the cytoplasm, and whichever one we're looking for. So for example, this first one is A, U, G. So if it has the corresponding code, so A would match with U, U would match with A, and G would match with C. So now UAC is its anticodon code, and it's going to go searching in the cytoplasm for the amino acid that matches it. So you can be given a table like this. Any table can be a table of DNA, can be a table of mRNA. This is a table of tRNA. So if you look at the codes, the 3 bases, so the first base in ours is U. So we look along the first row for U. The second one is A, so in the first row, in the third column, I'm now looking there, and the last base is C. So if I look for C across, I find that this matching amino acid is tyrosine. So this little tRNA is going to go in the cytoplasm, and it is going to pick up tyrosine on the one end. That's then going to come in to the ribosome, match with its codon, and it's going to stay there, while another tRNA that's sitting in the cytoplasm has another matching code to the next 3 bases. So the next 3 bases is CCU on the codon. So matching with C would be G, G, A. So now, if you look on the table for G, so I'm looking in this row, then G, so in this final block, A means I'm looking for glycine. So, in the cytoplasm, this tRNA has attached itself to glycine because that's its matching one. And it will come through and it will match along, and the ribosome will hold those in place together. Now it's holding them in place so that they can form a peptide bond to each other. That's going to be done with the help of the enzyme peptidyltransferase. Once it's formed its bond, this tRNA detaches and it goes back to find the next same amino acid so it can bring it in again. The ribosome then moves along to the next base while it holds that in place so that the next one can come in. And then the next tRNA is going to bring another amino acid, hold it in place, they can bond, and it moves out until eventually it gets to the end, and all we're left with is a polypeptide strand. So the whole process starts with what we call a start codon, and it ends with a stop codon. And once the ribosome gets to the stop codon, then it detaches. All of those molecules that we use, like the mRNA, gets broken down and reused for the next protein, and I have my polypeptide that will become the protein that I want. So that is the whole process of protein synthesis.